Section 1. A new kind of war. Can a virus be a cure? Can a virus cure cancer? This sounds like science fiction, not modern medicine. For decades we've viewed viruses as enemies, invaders, hijackers of our cellular machinery, our medical strategy, eradicating them, developing vaccines to prevent them, creating antivirals to fight them, yet a remarkable twist of scientific ingenuity. Researchers are turning this ancient foe into a new ally in the war on cancer. Imagine the battlefield inside the human body. Cancer cells are renegades, our own cells mutated and growing uncontrollably. They build fortresses, evade our immune system, and spread relentlessly. Chemotherapy and radiation, often like carpet bombing. Powerful, yes, but causing immense collateral damage. Now picture a different kind of soldier, a microscopic, highly specialized agent, programmed to seek out and destroy enemy cells, sparing healthy ones. This is the promise of oncolytic virotherapy. The idea isn't new. Over a century ago, doctors noticed something strange. Cancer patients who caught viral illnesses, like measles, sometimes saw tumor remissions. Rare, anecdotal, but seeds were planted. For years it stayed on the fringes. Too risky. But modern genetic engineering lets us disarm viruses, remove their ability to harm healthy people, and enhance their ability to target and kill cancer cells. A pivotal moment in oncology. Understanding the dance between a virus, a tumor, and our immune system. Scientists aren't just using wild viruses, they're designing them in the lab, turning a weapon of disease into a tool for healing, harnessing the power of nature, refining it with human intellect, deploying it with unprecedented precision. The question is no longer hypothetical, it's a real, active field of research, already changing patients' lives. Section 2. The 1-2 punch. How oncolytic viruses attack cancer. So, how exactly does this work? How can a virus known for making us sick become a cancer-killing machine? The strategy is elegant and relies on a powerful two-step attack. A 1-2 punch that cancer cells find very difficult to withstand. The first step is direct destruction. Scientists engineer these special viruses, called oncolytic viruses, to have a unique talent. They are designed to selectively infect cancer cells and then replicate inside them. Healthy cells have robust antiviral defenses that can fight off these engineered viruses, but cancer cells are different. Mutations that make them cancerous often cripple their antiviral defenses, leaving them wide open to infection. Once inside a cancer cell, the oncolytic virus takes over. It turns the cell into a virus-making factory, replicating thousands of copies. The cancer cell becomes stuffed to the breaking point. Then the cell bursts open. Oncolysis, or cell lysis. The explosion kills the cancer cell directly and releases a brand new army of virus particles, creating a chain reaction through the tumor. But that's only the first part. The second, and maybe more powerful step, is activating the patient's immune system. The chaotic virus-induced cell death acts like a giant flare, an inflammatory alarm. When cancer cells burst, they spill everything, including tumor antigens. For a long time, the immune system may have ignored these disguised cancer cells, sending "don't attack me" signals. Immune cells are drawn to the tumor site. T cells, natural killer cells, they find the newly exposed tumor antigens. The immune system is trained to recognize the cancer as a threat. The oncolytic virus unmasks the cancer, creating systemic, long-lasting anti-tumor immunity. The immune system is now primed to hunt down the primary tumor and any cancer cells that have spread. Metastasis. Section 3. The numbers don't lie. Evidence from the front lines. This all sounds promising in theory, but does it actually work in people? For years the evidence was limited to compelling lab studies and small, early-phase trials, but the data is now maturing. Researchers are compiling results from numerous clinical trials around the world. And the numbers are beginning to tell a very encouraging story. A major meta-analysis published recently in the journal Vaccines. This landmark study, led by El-Tanani and colleagues, pooled results from 36 clinical trials, covering about 490 patients with various types of advanced cancer. The first key metric is objective response rate, or ORR, the percentage of patients whose tumors shrink or disappear. When oncolytic viruses were added to standard treatments, ORR was nearly 3 times higher than standard therapy alone. For every 1 patient who responded to standard therapy, nearly 3 responded with an oncolytic virus in the regimen. This suggests a powerful synergistic effect. Another critical measure is overall survival, or OS, how long patients live after starting treatment. The meta-analysis revealed roughly a 16% improvement in median OS with oncolytic virotherapy. While 16% might not sound as dramatic as a threefold ORR increase, in advanced cancer it represents months, and sometimes years, of extra time. Importantly, this new weapon appears remarkably safe. Most side effects were mild: fever, chills, fatigue, and typically resolved within a day or two. Serious adverse events were rare, especially compared to traditional chemotherapy. El-Tanani et al. Vaccines, 2025. As with any cancer therapy, not every patient responds to oncolytic viruses in the same way. A key question for oncologists is, can we predict who is most likely to benefit? This is the goal of biomarker research. In virotherapy, scientists are homing in on two promising biomarkers. Tumor mutational burden, or TMB, measures how many genetic mutations are in a patient's cancer cells. High TMB tumors look more foreign. Each mutation can create a neoantigen the immune system can recognize. Low TMB equals a good disguise. High TMB equals a terrible disguise, covered in unusual features. Early data: oncolytic viruses often work better in high TMB tumors. The virus bursts cancer cells open, releasing neoantigens, a rich set of targets for the newly activated immune system. Second biomarker: the gut microbiome, trillions of microbes that help regulate immunity. A healthy, diverse microbiome primes the immune system. Certain compositions correlate with better virotherapy responses. Researchers are testing dietary changes, probiotics, and fecal transplants to turn non-responders into responders. Using biomarkers like TMB and the gut microbiome moves us from one-size-fits-all to precise, personalized medicine. We stand at the edge of a new era in cancer treatment. The future of cancer therapy may lie in precision virotherapy. Did you know viruses can fight cancer? It sounds like sci-fi, but it's real. They're engineered to hunt tumors. They infect cancer cells, multiply, and burst them from within. That chaos flips on your immune system to attack. Meta-analysis: objective response rate nearly 3x higher. Overall, survival up about 16%. Biomarkers like TMB and the gut microbiome matter. This breakthrough is real. Published in Vaccines. Learn more. Precision virotherapy is rising. El-Tanani et al. 2025. Efficacy, safety, and predictive biomarkers of oncolytic virus therapy in solid tumors. El-Tanani et al. Vaccines, 2025. Oncolytic viruses work via a dual mechanism: direct oncolysis and immune activation through antigen release and inflammation. Engineering confers tumor selectivity while sparing normal tissue. Systematic review and meta-analysis followed PRISMA guidelines, 36 trials, approximately 4,190 patients across solid tumors. Outcomes included ORR, PFS, OS, and safety. Standardized extraction and quality checks. Addition of oncolytic viruses yielded nearly a threefold increase in ORR. Median overall survival improved by roughly 16%. Progression-free survival signals were supportive where reported. Safety profile: predominantly mild, transient flu-like symptoms. Serious adverse events were rare. Tumor mutational burden correlates with immunogenicity. Higher TMB often aligns with stronger responses to virotherapy. Gut microbial composition influences systemic immunity and may modulate virotherapy efficacy. Prospective studies are exploring dietary interventions, probiotics, and fecal microbiota transplantation. Key challenges: delivery with immune clearance, stromal barriers limiting intratumoral spread, solutions under study, cell-mediated delivery, intratumoral injection, stealth design. Manufacturing at scale requires robust GMP processes and quality controls. Precision virotherapy: match-engineered virus to tumor biology and patient-specific biomarkers. Combinations with checkpoint inhibitors may amplify and sustain systemic anti-tumor immunity.